Perfect. So we're going to have a session with the microbiome, but focusing mostly, I'm going to provide an overview. What do we know about dogs and cats, the microbiome? And the main focus is really on function and specifically, how does it relate ultimately to cognition and brain disease and so on. So it's a very exciting session. I think there's a lot of new that we can see that we can modulate the gut microbiome and improve cognition and other diseases. So this is my Purina's disclosure slides. That's my opinions. For this, mostly I'm an employee of Texas A&M GI Lab. We offer microbiome testing, so we're going to talk about this a little bit. And then I'm also, thanks to Purina, now the Purina Pet Care Endowed Chain Microbiome Research, and our research is in part supported by the Purina Pet Care Research Excellence Funds. Okay, I think when you look back at the last 20 years, we knew that we have a new organ system that really, in the past, we kind of pretty much ignored, right? When we start looking at the GI tract, we need to really expand the view of GI physiology. The reason for that is because mostly we focused on this important part, the epithelium, the immune system, what you see here, and we kind of not realized that there's a huge other part of the GI tract, and it's really part of immunology or physiology. And so what we usually see is intestinal contents or feces. When you stain this with fluorescence, and there's a really beautiful picture of Chi Sung, one of our grad students, she did this take the colon from a healthy cat. You can appreciate how many bacteria are there. And there's a lot of variety, and because of that, the microbiome is really a functional organ. But it's also important to understand that this is just one part of the GI tract. When you look at this cartoon here, you have the microbiome, you have this mucus layer here, and epithelium, and the mucus layer here, that's in black, how important this structure really is, right? It's probably one of the most underrated structures in the GI tract because it really separates the microbiome or all the bacteria from the epithelium. And also important for function, I think, is, and also for dysbiosis, the gut is a very resilient organ. And that makes sense because you have all those changes here. We can induce dysbiosis with antibiotics, but as long as everything is intact here, I think there's a lot of capacity to counteract that. But over time, when this mucus layer degrades, we have inflammation, changes in the epithelium, then ultimately it's also going to change the microbiome. It's all together. We cannot really separate out one or the other. So I think it's important to understand dysbiosis can be a real shift in the microbiome. Dysbiosis is often probably a marker of some underlying change in the environment. But the other thing I was going to tell you that you don't always have to have a shift in the microbiome. And we're going to talk later about epilepsy and things. We don't really see a noticeable shift in the gut microbiome, but what can happen is the substrate that is available for bacteria, that can have an influence, right? So we don't always have to have a shift in something, we have a normal ecosystem, but if the wrong substrate because of diets comes in the lumen, it can be converted in other compounds and also have effects. So that's important too. The other important thing is that we're still realizing or recognizing that bacteria are everywhere. And this is a beautiful example. This is also Paula Giaretta, who was also a PhD student in our lab, and now she's actually a pathologist, also in our lab. And what she did is, her goal was to describe bacteria along the GI tract. And when you look at the crypts of a healthy dog, you can see that pathologists look at this every day. And then in the middle, they think, oh, that's like a mucus layer here, right? But if you stain this with silver stain, suddenly you see bacteria appearing in here. And when you do a fluorescence stain, you suddenly see that those crypts are full of bacteria. And those are mostly like helicobacter-like organisms. And in the GI tract of dogs, they're completely normal. And surprisingly, they're actually disappearing when you have GI disease. So again, bacteria are a part of our physiology. In most cases, they're normal. And usually when they disappear, the function disappears, and that really has often an effect. So often, loss of function is a more crucial aspect. But also I'm saying it's important to understand that as those bacteria, when we see them, we don't always have to see right away the GI disease, right? Often we look at this in GI disease, we look at biopsies, look at bacteria attached, we think it must be the cause, but they're actually simply there. So we have to really disentangle a little bit what is the cause effect of those. So I'm going to talk very briefly. So my goal is really to kind of provide a little high-level overview, what do we know about the GI tract, especially in dogs and cats, a little bit about the major metabolic pathways, how do we look at the microbiome in dogs and cats, what do we know about the changes, And ultimately, what actually causes major changes? Because I think it's important to understand, is the microbiome shifted? Or is the microbiome more or less the normal range, but then it's a multimodal factor? Then it depends on substrate availability and other factors inside better recognized for health. So we always hear that the microbiome is a metabolic organ, and I think that's probably really correct. Bacteria need to eat, they are hungry, and if they eat, they also convert things in metabolites. And those metabolites are ultimately getting absorbed by the host. And that happens along the entire GI tract. Now, there's probably many different metabolites available, but when we focus on major ones, some of them we know very well, like short-chain fatty acids, bile acids, and sometimes then we can really probably already establish a major of those metabolic pathways that probably play the major role. So like we know that short-chain fatty acids are important, they come from fiber, that's why fiber is so important. They get fermented by bacteria into short-chain fatty acids, and ultimately they regulate a lot of different functions inside our body, like blood pressure. They're important for myocardial repair, immunity, and so on. We know bile acids. We're going to hear actually along this talk much about bile acids, how they're actually also important in humans at least, or seem to be important in humans at least, in longevity. So there are associations between longevity and some of those bile acids. But ultimately, bile acids is a beautiful example how the microbiome is part of physiology, because bile acids come from the host as primary bile acids, and then they are converted by bacteria into secondary bile acids. So we need the bacteria to do this, and those secondary bile acids, or the ratio of both, is really important because they're signaling molecules. They affect many of those receptors. And again, they modulate lipid metabolism, glucose, insulin ratios, and also inflammation. And so here's an example that the conversion of bile acids by the microbiota exerts a lot of different functions. There's a bile acid receptor along the entire GI tract. And ultimately, as you can see, importance for glucose homeostasis, lipid metabolism, motility, energy, immunomodulation, and so on. Oops, sorry. And again, we also see the same in dogs. Also, Paula's PhD work was to really describe bile acid metabolism. So she had this beautiful, she took healthy dogs and stains with antibodies against the TGF5 receptor. And this TGF5 receptor is a receptor that gets activated by secondary bile acids mostly. That's the major agonist, and ultimately exerts a lot of those immune functions I saw you before. And you can see those receptors are everywhere. They're along the GI tract, they're in the goblet cells, in the macrophages, enteroendocrine cells, ganglion cells, and so on. They're also in the pancreas, so you also looked at the pancreas. Again, this bile acid metabolism is very crucial for health, not only in the GI tract, but to signaling in many other organ systems too. Another very important pathway, and one of the major pathways, is the whole tryptophan-indole pathway. And why is it also important? It's because tryptophan is a dietary amino acids that comes out of the GI tract, and the bacteria, it gets converted into serotonin. So serotonin is an important neurotransmitter. That's why the gut is so important in the gut-brain axis. But also what it makes it very important is because tryptophan gets converted by bacteria into indole derivatives, indole metabolites. There's a lot of them. And again, those metabolites in a certain range are also very important for regulation of the immune system, for regulation of intestinal barrier function, and so on. And when you have GI disease or GI inflammation, then two things can happen. You have GI inflammation, cytokine production shifts this whole pathway from tryptophan away from serotonin, but in the direction of chymothyretic acids. In addition to that, often GI inflammation goes hand in hand with dysbiosis. The dysbiosis is extreme. Bacteria that converts tryptophan into indole change, and ultimately we have both. So we have different shifts in them. So as you can see, this combination of GI tract, what happens in the GI tract, of the diet, and ultimately microbial metabolism, depending on health or disease, plays a major role in those pathways. And here's an example. We did this in the past. Many years ago, we looked at what happens with metabolites along the GI tract. And you can see in healthy animals, and you can see beautifully that you have a lot of tryptophan in the small intestinal tract, but then ultimately we can measure very, very little in the colon rectum. And the reason for that is again, as you can see, most of it's probably going to be absorbed, but then whatever goes into gets converted by bacteria in those indole triacetates. And we also know that, for example, if you have GI disease, you can actually measure tryptophan increased in fecal samples. That means what happens is you have lack of absorption, but also lack of this, through dysbiosis, lack of conformation, and so on. So again, two things can happen. So we need to also keep this in mind. Inflammation and dysbiosis often go hand in hand. So when we look at the microbiome, I think we learned a lot about the last few years. There's a lot of bacterial species present, but we did several studies now, and that's also very consistent with other authors, is that ultimately, yes, we can identify thousands of species across many healthy animals. But if you look at which bacterial species are really present in every single individual, it's actually not that many. In this study, those are 12 healthy dogs. In green is present, in red is absent, and you can see there's really only like 25 bacteria present that are really present in every single healthy animal. If you go a little bit deeper, there are 36 in 9 of 12, and so on. So again, all I'm just saying is that some of the core species, they are probably also from the function, seem to be the most important one, right? So those are the ones that probably also have the most metabolic pathways. Now, many of those lower abundant species or lower prevalent species can be important and they can have certain functions, but again, focusing on the major one can bring us a little bit further into characterizing normal abnormal gut microbiome. And looking at those are bacterial species that are really highly prevalent, and those are the ones that in all the studies that we did over the last 20 years almost, always keep repeating. It's Clostridium rhamnosus, that's an important bile acid converter, Faecalibacterium, that is very famous or known already in human medicine, also in veterinary medicine. So again, we have some core bacteria there. And so when you look back, and we have this dysbiosis next to many of you are familiar with, and in the beginning we really looked at, it's a shift in the gut microbiome, normal, abnormal, but over time we've refined it. And so this index measures actually really 7 of the keystone bacteria. 5 are usually that we consider being better, or they're mostly normal. And some of them, they're harmful. Harmful is probably the wrong word. I need to change this slide finally, but they're usually overgrowth bacteria. So when something goes down in an ecosystem, usually something overgrows, right? And now we have reference intervals for those bacterial taxa, so we can measure reference intervals, and we can better kind of estimate what is the prevalence in healthy and diseased animals. And so ultimately, that also means that we can characterize the microbiome in 2 different ways. Usually we have an index below 0, where most healthy dogs are gonna fall. So we call it kind of normal. We can really completely decide what is normal, but at least most healthy dogs fall in here. And then we have some bacterial taxa that are, some samples or some dogs have an index below 0, but at least one of those taxa is out of reference interval, then we define as a minor change in the microbiome. And then we have 0 to 2, which is a moderate shift, and the more the microbiome shifts, the more the index goes up, the more the microbiome shifts. And that's kind of important because also clinically, we kind of more and more realizing that a major shift in the microbiome is usually associated with chronic GI disease, and usually something that's persistent. Yeah, we can bring, even FMT, we can bring it back to some degree, but often relapses. And I'm going to come back to this one. But a more severe shift in the microbiome is definitely a predictor of persistence in those changes, and often associated with more relapse and so on. And recently, we kind of did a big study. And so what we tried to do is really assemble a data set of roughly 300 dogs of different phenotypes. So in this study population is healthy dogs, dogs with acute diarrhea, dogs with epilepsy, dogs with non-GI neoplasia, and then we have dogs with chronic enteropathy. And what you kind of see that the index itself, so again, when we target 7 of those key bacteria, predicts pretty well how the whole shift of the microbiome happens based on shotgun sequencing. So samples in green, are normal, so they have an index below 0 and all taxa within reference interval. Yellow is minor changes, so index is below 0, but some taxa abnormal. And then you have the 0 to 2. You can see already it shifts away a little bit. And then blue is everything above 2. And while I'm not showing this one, but I also can tell you that we can predict pretty much well the severity of those shifts. So dogs here usually have an index of 4 to 5, and those dogs that are here, they have an index of 7, 8, or 9. So as you can see, when you really appreciate data from itself, this is a huge shift, right? So an ecosystem, it's not going to shift by itself, that's easy. Something major has to happen in environments. And I'm going to come back to this one later when we say, how can we fix the microbiome? Usually we cannot fix it anymore. Even in fecal transplants, those dogs here are usually going to persist forever and so on. And so that's important because often when you see what is a shift, it's very difficult drastically. But as you can see, even 0 to 2. I also want to highlight this, for example, dogs with epilepsy, they are somewhere here in this range. That means they don't really have a shift in the microbiome, but doesn't mean there's no some metabolic pathways, some small things, substrates available and so on. But it's important to differentiate between those different phenotypes. And again, even in this, we had 76 healthy dogs, and you can see that only really 17 species were present in every single animal. So this, I guess, this core microbiome is quite consistent across the studies. So, helping with this, it helps us two things. We can look at untargeted methods. We can identify small changes. We can know a species, but also having the index helps us to say how much does it actually compare to a reference interval, to the whole population out there. So it allows us to get much better shifts in the microbiome. And so when you look, for example, in puppies, and I think that's a very consistent picture. I only have a slide from dogs, but similar things happen in kittens and also horses, that when animals are born, they usually are not born without microbiome. There are some papers out there that have shown there's a fetal microbiota, but there's actually very strong suspicion now that this is probably more through contamination. Just recently, a few months ago, a paper came out in Nature kind of trying quite convincingly to say that the fetal population is probably more like a spurious contamination and so on. So generally we are pretty sterile when we're born. But really, within a few days, when you look at qPCR, within 2 days, suddenly we have a lot of bacteria. But you can see the populations are different because at day 2, they cluster here, and you can see afterwards day 21, day 42, and day 56. The 56, they're coming pretty close to the dams already, right? So there's a huge change in the microbial composition over the first few weeks of life. And you can also see the same thing in diversity, which really goes up exactly by age and so on. But interesting, at day 56, the puppies are still just slightly different from their mothers. We look at the dysbiosis index now. That helps us to see actually a reference population based. We see the same thing. Again, in the beginning, we see a lot of environmental bacteria, E. coli, because again, the gut doesn't have yet the normal flora. So we're taking on everything from the environment. So you can see those dogs have a lot of E. coli. At the same time, they have very, very little, almost none, of the normal anaerobic flora. But pretty rapidly, and that really correlates usually with weaning. Not that weaning is the major factor, but you know, the GI tract progresses in function. After we have solid foods, pH changes, enzymes, you know, the epithelium grows and so on. That makes the gut much more normal for the anaerobic flora. And you can see nicely how E. coli decrease, then expand pretty much in the reference interval in adult dogs, and the anaerobic bacteria really become normal. And that's why also the dysbiosis index is at this age physiologically increased, right? Because they don't have yet the same flora that adult dogs have. So you can see it's increased, but it correlates very nicely, 7 to 9 weeks, and then somewhere in this range of 10 to 16 weeks, and most dogs are going to fall in normal. So it allows us really to track actually individual dogs, how they fall in the reference interval. And again, what is very, very important to understand is that the microbiome controls also the presence of other species. And we have always this discussion, what is an enteropathogen? Clostridium difficile in humans is an important pathogen. In dogs and cats, we don't believe it is one. And this is an example. Those puppies, completely healthy, no diarrhea, nothing, have a lot of Clostridium difficile at the first few weeks of life. They also have a lot of perfringens in the first few weeks of life. And you can see suddenly, at this week 7 to 9, there's a drop without really any specific intervention. And why is that? It's actually very simple, because Clostridium rhamnosus, which is this important bacterial group that converts bacterial bile acids, exactly comes into this reference interval in most dogs at this age, okay? And Clostridium rhamnosus is a bile acid converter. So you can see in the early age, they don't have secondary bile acids, but when rhamnosus comes at 7 to 9 weeks, you have this conversion from primary to secondary bile acids, and we know that those are very important regulators of Clostridium difficile. Secondary bile acids can really pretty much inactivate Clostridium difficile or completely exclude them. And that's important because I think we still have this notion that there's a lot of enteropathogens, right? In the US, I think a lot of puppies and kittens receive antibiotics because there's perfringens, they have diarrhea, we check in the enteropathogen panel, we see some of them and they get a lot of antibiotics, right? But in the end, this is a normal transition phase in the microbiome. A normal microbiome controls the presence. So those are all overgrowth bacteria in dogs and cats. Over lifespan, over adulthood, obviously we don't have really many studies, long-term studies, but generally we have enough studies, I think, to already suggest that there is actually not major changes. Diet is one of the most important regulators of the microbiome, but diet doesn't really change a huge shift. It doesn't make any sense that it would shift it drastically. Diet is an important substrate, modulates some of those components, so that's important. And so usually when you can put them on different diets, we see some significant differences, but they stay usually within a huge range of normal. As you can see also Clostridium melanonis. Same thing in cats. This is a sampling that we did in healthy pet cats, 18 healthy pet cats, multiple time points over 2 months. Some cats have been sampled almost like 30 times in this time span. And you can see there's a difference. Some cats definitely have a baseline difference, correct? Some cats are very, very stable in the microbiome. There's almost no difference. They really cluster very tightly. Some other animals, you can see the microbiome actually varies in this time frame quite drastically. And that's a question we don't know. Can we regulate? Is it more important to be stable? But generally, I want to say is that it stays within this huge range of normal. And also in cats, we see that the increased dysbiosis index, I'll come back to this one, is mostly when we have chronic GI signs. We have this increase, and the populations seem to be very similar between the USA, and there's a recent study in Italy where roughly like 75% of cats with chronic GI disease have an increased dysbiosis index. Interestingly also, when you look at just simply back some animals, we have some animals that we sampled over many years, some of our from our students or technicians. And some animals are really fascinating. They stay really almost in the same range, but some of them vary still quite a lot. So I think there is definitely a difference between why they vary. That'll be interesting to see how can we actually nutritionally or something like keep them stable. So coming back, what are the major changes in the microbiome? And that's really 2 that we know of at the moment. There's maybe some other ones, but at the moment we know it's GI disease and antibiotics. And this is really something that obviously we just described a few years ago when everybody thought, well, we give metronidazole to treat dysbiosis, right? That's still in the many textbooks. Now we know pretty convincingly that antibiotics, especially broad spectrum ones, definitely induce a severe dysbiosis. It's different between different antibiotics. It's also different between species. For example, we know that metronidazole makes a huge dysbiosis, and what happens is that in some animals they come back pretty quickly, but some animals, like here, they're still going to be abnormal even 1 month after end of antibiotics. Amoxicillin, for some reason, in dogs doesn't lead to major changes, but it's really, really bad in cats. Cats are extremely sensitive to almost all antibiotics, and they usually stay also very long-lasting. And you can also see that some of the important bacteria, like Faecalibacterium, short-chain fatty acids, are to different degrees dependent on what antibiotics do. And again, Clostridium melanonis, which is one of those important bile acid converters, as you can see in many dogs, it stays abnormal, long-lasting, and so on. Again, those are healthy animals, and I said before, if everything else is normal, there's a buffering capacity, right? But long-term, probably, it's definitely not a good idea if somebody stays in a dysbiotic state. And that's really data from humans that we know now. And in humans, it's, I think, very, very convincing. There's many, many studies out there that associates antibiotic use, and especially broad-spectrum antibiotic use, to some of the chronic diseases. In this case, it's childhood asthma, rhinitis, dermatitis, but there's also many studies in human medicine linking broad-spectrum antibiotic use to development of inflammatory bowel disease and so on. Important to highlight, it's a risk factor that's still pretty small, So it's not that means everybody whoever is antibiotics is going to develop the GI disease, but nevertheless, it is there, so we need to be kind of aware of it, that changing the microbiome, when changing antibiotics, also changing many of those metabolic pathways, and ultimately we have this risk factor, then becomes really cost-benefit ratio, yes? What is the beneficial for this patient, and do we need antibiotics? And I think we all know now that in GI disease, in many, many cases, we don't need antibiotics, even not in the most cases. What do we know about cats? This is a study from Eva Stavrolaki. She did a sequencing study first, and then we had actually the chance to run the dysbiosis on her samples. And what she did is she looked at kittens. She enrolled them roughly at 2 months of age. And you can see at 2 months of age, all of them are still, as expected, like in dogs, they still have a little bit increased dysbiosis index because the microbiome is not matured now. If you look at the groups in red, that's the control group. You can see that after 3 months, 4 months, again, almost all cats are below 0 and a few cats are here. But then she also enrolled 2 groups of kittens at 2 months of age that received antibiotics because of a respiratory disease. And you can see that the doxycycline group didn't really change, a little bit more delayed, but it was not significantly different. But you can see definitely that amoxicillin clavulanic acid definitely in this age really slowed down the maturation of the microbiome. It's caught up, as you can see, and then it was not normal. But very interesting is that there is a study now, a follow-up study from this data, and there's an abstract submitted to ECVM that actually suggested or showed that kittens, when they receive antibiotic use in this young age, have an increased risk for chronic diarrhea later on. So that's the first kind of evidence that also what we could see potential antibiotic use and risk of developing GI disease later on. So I told you a little bit before, acute and chronic. What changes? What is very interestingly actually is that we definitely see different patterns in dysbiosis. Dysbiosis, an increase in dysbiosis index, and especially this loss of coccidioidonosis, is mostly seen with antibiotics or chronic GI disease. It can be in any form, because we know now chronic enteropathy is probably not a spectrum, it's probably it's a spectrum of different diseases, the same disease but different manifestations or treatment responses. But interesting in acute diarrhea, and it's we have several studies now that usually you have only a minor change, and if you have an increase, usually it's transient. And that was kind of interestingly is because, again, we expect a dysbiosis in everyone. And so we have 2 studies now, both from Munich, so thank you for the collaboration, Stefan and Kati. But you look at the model of AHDS, so acute hemorrhagic diarrhea syndrome, we have this acute onset of this severe necrotic enteritis. But we also know that this is a disease that usually recovers pretty quickly with some symptomatic treatments. Usually it recovers. Dogs are usually after 7 days to 10 days have no signs anymore. And what you see that very few of those actually have an increased dysbiosis index. And then day 7, day 14, it really decreases. One study from 2018, and the same thing we see in another study now from 2022. A little increase, but then actually the microbiome normalizes very rapidly. And it really correlates very well with the clinical improvement, and also correlates very well with NET-F toxin, which we believe is one of the major inducers of this disease. It was confusing a little bit, because this necrotic enteritis, you would think that this would actually cause a severe dysbiosis, right? But again, what we're doing now too is trying to find markers that tell us how the function, or not the function of the GI tract is, or the stability. So we're measuring fecal cell and fatty acids. And we look at those dogs in HDS, so those are fatty acids and sterols. Some of them are phytosterols that are markers. They're part of cell membranes, and they're supposed to be absorbed. They can give us a little bit of hints, absorptive capacity, and also shedding or damage of the GI tract. And what's kind of interesting is, you can see at day 0, we have day 0 of those dogs, they look completely different, like those healthy dogs, right? This is increased because it's red, and the same are very decreased, and so on. So there's obviously a huge difference at day 0, but it's kind of amazing if you follow them up, day 3, day 7, day 14, day 21, the gut really, really actually heals pretty quickly. So even after 21 days in this acute necrotic enteritis, they become very, very close to normal. And again, for me, it kind of makes sense. It's almost like an acute onset of a damage, but if everything around this still works to some degree, pretty much the gut can recover, right? However, we know that those dogs are in increased risk for developing chronic GI disease now. So there's still definitely some damage potentially that is, but generally at the acute moment, the healing is there. And this is important because in chronic GI disease, we don't see that. In chronic GI disease, once we see change in the microbiome, it usually persists, okay? And probably because we have those changes, those chronic changes in the function of the epithelial tract. So it's not just that there's a dysbiosis, but dysbiosis is probably the reaction that there's some major change in environment. And we know when we look at the biopsies, for example, we have villus atrophy, villus blunting, the brush border is reduced, and so on. So the function is different, and that really says chronic. And so this persistence, and we see this persistence in dysbiosis. Now, we can induce remission in those animals. We can manage them, but the expectation in most cases is that they're never going to recover completely in the function. And so it becomes a very different treatment approach, and also important to understand that often those patterns can be already there before disease comes. And so that is important because we look at different organ systems. We need to understand, is the gut functioning normally? Or is there some comorbidity that is probably not manifested in clinical pictures? But we know now we can manage them, but often when they have dysbiosis, this is going to be persistent, present, because the gut function doesn't recover. And so I want to talk a little bit about fecal microbiota transplantation, or FMT, because for the sake of time, this is probably the gold standard. So when we look at what can we induce with changes, it's going to help us to really understand expectations. And then later we can hopefully modulate it and reduce it to some of those pathways. So that's the future. And so what was very interesting is that we already have plenty of case series, case reports, and so on, that we see some consistent patterns. One of them was interesting, that Jennifer Scheitman, who is kind of the pioneer of fecal microtransplantation, she did this in Manhattan for many years. We did this kind of, there was published in SVM abstracts a long time ago, and discovered is that, which back then was actually surprising. Back then we thought that if you have dysbiosis, FMT should be really able to reverse it, and that's the proper indication. What we saw is that dogs with this severe dysbiosis here, even after fecal transplant, many of them don't recover at all. Or if they recover, they usually come back. So we couldn't really change the microbiome even in those dogs with severe dysbiosis. And those dogs usually also relapse pretty quickly. On the other side, if you look at dogs that are here, that are more like this in this mild to moderate dysbiosis or in the normal range, the microbiome recovers pretty quickly, and that's also associated with a better long-term response. And that's kind of also confirmed by Linda Thorsten in her retrospective study. When we have severe dysbiosis, those are usually poor responders. They respond maybe like for a short term, relapse pretty quickly. We have dogs that have more milder form of dysbiosis. They are usually the ones that respond better. They still can relapse, okay, but the response rate is usually longer and so on. So again, the severity of dysbiosis is really an indicator. It actually makes sense now, because probably it reflects much more changes in the mucosal environment, right? And that means that's why we cannot change. So those are dogs that are, remember, when you saw them, those are the really huge on the right side. They're very far away. So there's really something major in the environment. And you can see this. This is like a case report where you have a dog, has severe diarrhea, receives an FMT, it looks better after 2 or 3 days. We can improve the dysbiosis index, it looks all good, but then, as you can see, after a while, expectation is that it's going to come back. And again, most likely reflecting this persistent change, dysfunction of the GI tract and so on. On the other side, and that's important because, as I mentioned before, antibiotics are commonly used, right? And for many reasons, they are used properly corrective. So we need to always use antibiotics. But we also want to see in the future, how can we actually bring the microbiome back? And this is a study from Jamie Hu and Frederick Gushen. They was presented as an abstract. And what they did, they looked at healthy cats in a colony, and they gave them amoxicillin clavulanic acid for 10 days. And you can see all those cats stay here normal. After 10 days, all of them become dysbiotic. And then those in black here, they received just nothing. So they were just watched over time, and you can see the recovery of the microbiome takes much longer. And in some cats, it's still there 6 weeks or 8 weeks post-FMT, so a long term. Versus those in blue, they receive 1 day after the last antibiotic treatment and FMT, and you can see pretty much all of them recovered pretty nicely quickly. So again, it depends really on the underlying process. If we have antibiotic dysbiosis and the gut function is probably normal, we can recover them pretty quickly. And that's why it's important, because I think when we look at risk factors, I think we not going to do long-term FMT in every cat, so we need to find modalities we can bring back, but it is important to understand that there's probably a lot of subclinical dysbiotic animals out there. This is an example. For example, Kobe is the cat of one of our technicians, and we did healthy cats for a study looking at the microbiome. And so she was very excited because her cat is super healthy, has no GI disease, has never received antibiotics, to her knowledge or to her memories. And so we run a dysbiosis index, and you can see it's actually normal, but it's really missing Clostridium melanonis. Okay, so this is important bile acid converter. And that's really only 2 reasons, GI disease or antibiotic use, right? And so we asked her, and she said, no, she doesn't remember antibiotics. And we asked her again, she doesn't remember antibiotics. And after just chatting about something else, she said, oh yeah, 2 months ago, her cat received cefazolin for skin disease. So it's important that we understand a lot of animal owners forget about that. So we said, okay, that may be a nice indication for an oral FMT. We gave her 1 capsule, 85 milligrams of lyophilized feces. And you can see that here noise comes slowly, keeps coming back, but kind of relapsed again. So we decided let's give another week of 2, and you can see then we can bring the microbiome back. And then even 40 days after FMT, it still was normal and so on. So it's possible to bring the microbiome back. FMT is a very crude method, obviously, so the goal would be afterwards in the future to find a nutritional solution, how we can bring it back. But I think it's important that we can recover some of the function of the microbiome in animals that stay dysbiotic for a long time. And again, just as a final slide to just bring you a little bit that, again, it depends also on the GI disease. This is a case of a cat with chronic GI disease. It has been already for many years treated with prednisone that controls vomiting, but the stool quality was really, really bad. So the owner said it stunk so bad that I took it outside. Index is abnormal, so we decided FMT. And you can see that actually 2 days later, 19 days later, the stool quality looks better. Not perfect, but better. Index recovers a little bit, but the moment we stop FMT, the microbiome dysbiosis keeps coming back and so on again, this chronic dysfunction. So understanding that whatever happens in GI tract, sometimes you're going to have to treat those microbiome management for a long, long time. Okay, so that's kind of the major factors in old age. And just a few slides about microbiome in old age, and that's really not much information out there. So this is kind of new studies. I think there are a few studies out there, but what is discovered that in young age, the very few in dogs and cats, the first 2 or 3 months of life, huge changes. I think in humans, it's probably the first 2 years where the changes. But later in aging, we don't really expect a major change in the gut's microbiome. There are some studies that have shown that there are some factors, but it's probably not necessarily an aging step itself. It's probably depending on many lifestyle factors. Older people have limited fiber intake. Obviously, we have more antibiotic usage. And ultimately, you also have a lot of comorbidities, correct? So that all can affect the gut's microbiome. Interesting, though, is, like we heard before, centenarians. There are definitely studies now that have looked at, are they different? And what they have seen that, for example, centenarians usually have decreased susceptibility to aging. They have less chronic inflammation and less infectious diseases. And what is interesting, and that's why coming back to this bile acid, which seems to be really an important pathway, often unfortunately affected by antibiotics and GI disease, but they have identified a few isoforms of secondary bile acids. So again, bile acids is really a complex pathway. So there's I don't know how many. I mean, we have a new extended panel with 30, but I think there's probably like 50 or 70 bile acids out there. So still of much to learn. And so different bacterial species can still metabolize some of those even secondary bile acids. And so again, it's not really clear, it's an observational study, but there's definitely some association that this potentially could reduce the infection with pathobionts, because we know that bile acids are important in regulation some of those pathogens. Another study is the viriome, which is something that is really not really well described. Obviously, the gut's microbiota is not just about bacteria, while 95% of them is bacterial, but we still have viral, we have bacteriophages, we have other organisms. And also in this study, they have looked at the viriome and also shown differences. And they also looked at the function and ultimately suggested or expected as an increased potential for converting methionine to homocysteine, sulfate to sulfide, and so on. So all pathways that maybe support the mucosal integrase. So we don't expect major changes, but maintenance of microbial function. In dogs and cats, really, there are some studies out there, but obviously everything changed since molecular tools and more. Obviously, we always develop new technologies, and so some of those studies, while they found some decrease in lactic acid bacteria, it's difficult to judge how many other changes were there because they focused really on specific little group. But again, overall, all those changes also, similar as in humans, seem to be rather small. We have just a study that is just in submission to a manuscript, but they have shown, in this study, the authors have shown that dogs that are more older than 7 years had a statistically significant shift in the microbiome. However, if you look at the size effect, it was very small, again, a very minor shift. And it was also confirmed by this dysbiosis index that the proportions of dogs at an index above 0 was very similar between adult dogs and senior dogs. And that's the same proportions we usually see in every single study. There's this proportion of animals that we believe have subclinical dysbiosis. So ultimately, I think there is something going on in age, but it's not really a strong effect, and then it's going to be a metabolic factor. But ultimately, I think that's actually good news, because I told you before that if most dogs fall in this range, and all our data from different studies, FMT studies and so on, would actually suggest that we want to be in this range when we're aged, because a normal more or less a normal system is easier to manipulate, right? We have potential solutions with nutrition and so on, with other factors to maintain it, versus if we're here or here, that probably indicates that we also have underlying GI disease, and that's a different phenotype. We have to treat two different things: changes in the mucosal level and also age. So in summary, rapid changes we have in the first few of lives. Then somehow at this age of, let's say, 3, 4, 5 months, maybe there's some smaller indications. Usually we don't have the strong data when the microbe is fully mature, but I would think it's probably between 6 to 12 months. During adulthood, it's relatively stable, but we know there are definitely factors that can change the microbiota. We can, especially antibiotic use and GI disease. And again, in older age we have subtle changes that are likely due to those factors. That's ultimately actually is not a bad thing, because again, I think in a huge range of normal, we can modulate it much better with multimodal therapy approaches, so that's the exciting news. So it's going to be probably dietary interventions with additions of bacteria and so on. But if we have shifts in the microbiome that are drastic, then probably we have a secondary disease process. So that means that's a different underlying reason why the microbiome shifted. So with this, I would like to thank many of my collaborators and also students from our GI lab, all my students, and then ultimately many collaborators from different parts of the world that can help us providing samples that we can really look at many different of those, and really get a global picture of microbial shifts.